Okay, we can get started. Somebody or a couple of people online, could you indicate whether or not you can hear me and or see the slides, please? Perfect, thank you very much. So I realise I'm not visible to the people online, but I am standing in the corner and I will check afterwards for questions for the people online. Thank you for confirming that. So, hello, I'm Rona Strawbridge. I've been working with genetic data to understand complex diseases for about 15 years. I'm now a senior lecturer in mental health, and I am in the building most of the time. So if you want to talk about this kind of stuff at any point, give me a shout. I was going to say something else, but I can't remember what it was. Ah, yes. I've been working with not only genetics, but international genetics consortia for a long time. So when I'm discussing some of these concepts, it's not just my opinion, these are field accepted standards. So the overview of the talk, what are polygenic scores? How do you decide which variants go into them? How are they calculated? How can you use them, and things to consider that are important. Firstly, terminology. Genetic variants incorporate lots of different types of genetic variation. Single nucleotide polymorphisms, copy number variants, insertions, deletions, and mutations. Polymorphisms are specifically a genetic variant where the minor allele frequency is greater than 1% in the population. A mutation, again, is specifically a variant that is at less than 1% minor allele frequency in the population. Frequencies of variants are very much specific to ancestry, so always just find the ancestry of the population you're talking about. What is a polymorphism, so greater than 1% in a population of African ancestry, could well be a mutation, i.e. less than 1% in a European population. So defining which population you're working with is very important. When we talk about alleles, we're talking about the different versions of DNA at any point in the genome. The major allele is the one that's most frequent, the minor is the one that's less frequent. And again, that can change between populations. When we're talking about a genotype, we're talking about the scoring of the alleles at a given locus. So if your alleles are A or T, then the genotype can be AA, AT, or TT. RSIDs are the unique identifier of genetic variants, and it's very frequent that you see something like this written. You've got the RSID, hyphen, and then the allele, and that just defines which allele you're talking about to make it easy to understand. So, polygenic score or polygenic risk score? There is no agreed upon description. Polygenic risk score makes more sense if you're talking about risk of a disease. Polygenic score is more generic. I am too used to using polygenic risk scores, so I will stick with using polygenic risk scores. For complex diseases, many genetic variants influence each trait or phenotype. Each variant only has a small effect, and each genetic variant affects, the effects of each variant are not in isolation. They are part of the whole. It depends a lot on the surroundings, and that is the other genetics in the environment, the other genetics and the environment. So a polygenic score or polygenic risk score is the combination of many genetic variants. The genetic variants can be across the whole genome. It doesn't have to be those that are close together. So how do you choose which genetic variant to include? Genome-wide association studies, these are the gold standard for how you identify which genetic variants are associated with a phenotype. So you could conduct a genome-wide association study of schizophrenia, heart disease, BMI, a biomarker. And if you then, the GWAS catalogue is a great place for looking for genetic variation. or which genetic variants have been associated with a phenotype. Most genetic variants are outside of the coding region. So this is an important thing for considering. We can also look at genetic variants that are associated with expression of a particular gene. We can find this information from the GTEx portal. So if we want to mimic the effects of PCSK9 inhibitors using genetic variation, we can identify all of the genetic variants that are associated with reduced PCSK9 gene expression. We can also choose functional variants. These are something that introduces a stop codon or changing in splicing. They are rare, but they can be combined with common variants. If you are looking just at rare variants or within a gene, burden testing should be used, not polygenic risk scoring. It's a slightly different method, but it's better suited for rare variants. So if you're calculating a PRS, you can include genetic variants across the whole genome, irrespective of the p-value for the association. You can take genetic variants that are only associated with the phenotype. So typically we'd take variants that reach genome-wide level of significance. That's a standard threshold for genetic studies. However, if you have a genetic study that is a very small number of individuals, you might not reach that standard genome-wide threshold, in which case it is possible to argue for using suggestive level of significance instead. Nominal significance can also be used. Depends a little bit on your question, the data set you're working with, and how you argue that. There's always a trade-off between power and specificity. So the more genetic variants you include in the polygenic score, the more powerful your analysis is going to be. But the fewer, if you have the whole genome, quite a few of those genetic variants don't have any association. They have null associations, so there's a lot of noise. So it's always a trade-off between specificity and how much power. I would say, though, that if you have less than 10 genetic variants in your study, don't bother. It's too low power to be particularly useful. If you use genome-wide study, you've got a lot more power, but then there is quite a bit of noise as well. If you're comparing multiple polygenic risk scores, you have to account for multiple testing as is standard for all analyses. Also, you only want to include independent genetic variants. If you include all the genetic variants in the region, you might be counting the same information multiple times, and that's not a good way. You're just going to inflate the results. It's not going to be a real finding. So, linkage disequilibrium is how you identify the genetic variants that you include or exclude by how often they are co-inherited. If you have a plot like this, each genetic variant is along the top. Here are two genetic variants. The black color shows that they are co-inherited all the time. If you include both of these variants in the polygenic risk score, you're counting the same information twice. So we don't want to do that. Same information is basically if the number is a percentage, greater than 80%, it's the same information. Less than 50% is different information. These are ballpark figures. So if we look at all of the ones that have very high inheritance, we can remove these. This variant represents this block. This variant represents this block. And then we probably include these ones as well. So to calculate the PRS, basically, we're converting lots of categorical variables into a linear variable. We count the number of risk increasing alleles. So if for this variant, the C allele is the risk increasing allele, we have 0, 1, and 2, because the assumption is an additive genetic effect. We assume that 2 copies has more of an effect than 1 copy, and that's different from 0 copies. You can use a dominant or a recessive model, but you have to have good biological reasoning to do that. You can just count the alleles, or you can count the alleles and weight the alleles by the effect size. The effect size being the beta or the odds ratio. For common genetic variants, the weighting has very little impact. I think commonly we weight them at the moment, but actually it doesn't make that much difference. However, if you're combining rare variants with common variants, the weighting is important because rare variants typically have a much greater effect than common variants. So we want to align the alleles in the same direction, typically trait increasing, and how you deal with missing data is important. Ideally, you only include individuals who have all the genetic variants. However, if you lose too many individuals, that's not the best approach possibly. So that's something to consider. So if we have these 4 variants, and this is the increasing allele for each variant, here are the genotypes, and here are you code them into how many alleles for each variant, and then you just add up the number of variants, the number of alleles, sorry. For those in the room, if you're struggling to remember this, the slide is being recorded, so you should be able to look at it later as well if you need to. So the polygenic risk scores can be calculated automatically using software. You don't have to do the manual calculation. Manual calculation is fine if you have 5 variants. If you have 300, it's not fine. So then you want to use some of the software that typically runs in R. Precise was an older method. You chose the p-values that you wanted to compare. That can be good if you know what you want to do. If you're not sure, it's not so helpful. Precise 2 automatically defines the best p-value to use. I'm not sure what their definition of the best is. That's something to read in their software, their manual. LD-PRED and LD-PRED2 use the whole genome. You require a reference data set for checking the LD between the variants. Those are major different methods at the minute. New methods are always being developed. So in 3 months' time, the most fashionable might be something else. It's definitely worth checking the distribution of the PRS. Most polygenic risk scores should be normally distributed. If you're including rare variants, that might be skewed slightly, but if it's not normally distributed, you might need to check what's going on and why. So we can use the PRS in a bunch of different ways. We can assess the impact of genetic risk for one phenotype on the risk of another phenotype. I'll show you 2 examples of that. We can use it for causal modeling using Mendelian randomization. We can use prediction modeling. I don't have any examples of that, but there are some in the literature, and I've got some papers at the end as reference. And we can potentially use it for risk stratification. Again, that's not something I've done, but there are some references for that. It depends a bit on which type of data you have. If you have individual level analysis, so if you have UK Biobank, you have all the individual data, you can adjust the models for all the covariates you want. Alternatively, if you're using summary level data from genetic consortia, you have their analyses and just the results. So 1 sample or 2 sample methods. So if we're looking at expression, PRS of expression versus phenotype, the question we asked was, does increased autophagy influence cardiovascular disease? So we identified genetic variants which regulate gene expression of all the genes involved in an autophagy pathway in a relevant tissue. We used the standard significance level that GTEx say for defining which genetic variants go into the model, checked the LD, created polygenic risk scores by gene, by pathway, and by tissue. Standardization of the PRS means you can compare between the different PRSs. And then we used these in a logistic regression model. Obviously, multiple testing is an issue there where we're checking a bunch of different PRS. We can use Mendelian randomization. So we use genetic variants as a proxy for lifetime exposure. and we can test the impact of exposure on an outcome. So genetic variants are used, so we could have BMI here as the exposure, genetic variants associated with BMI, and then we can test the impact on, say, heart disease or depression. There are some major assumptions for Mendelian randomization. There should be a strong association between the genetic variants and the exposure. So GWAS significant is very good there. The genetic variant should only influence the outcome through the exposure. Not always easy to test or verify, but with some knowledge of biology, you should be able to make a good prediction of that. And the genetic variants should not be associated with any confounders. Again, we can only check that for the confounders we measure, so there are always limitations on how certain you are that that assumption has been met. You can use individual level data, either in Stata using instrument variable analysis or using R. Summary data, so Stata is then much better if you only have a few genetic variants in your score. If you're using big data, MR-based or summary data methods in R are very good when you have lots of genetic variants involved. MR-based is a web-based program based on R that you can pull down from their menus the different phenotypes you want to check for associations with each other. I am not a big fan of it because I've tried the same analysis 3 times and got 3 slightly different results. I don't understand why. When I don't understand, I don't like it. So that's just a caveat. Be aware of that and check what you do a couple of times to see how robust it is. And statistical methods are all very well, but you have to consider the biology. Is what you're trying to do plausible? Are there important confounders that have been missed? So, as I said, we can look at whether PCSK9 inhibitors, we've had some evidence that suggests that they might have a negative impact on mental health. So to test further, we looked for genetic variants associated with reduced levels of protein or gene expression of PCSK9. Ancestry matching is important, so we found the genetic data from European ancestry individuals. We have European ancestry individuals in UK Biobank. The Psychiatric Genetics Consortium for the Mental Illness, UK Biobank results for other phenotypes associated with mental illness. Harmonization of the data is important, checking that you have the same genetic variants and you're talking about the same alleles and you've aligned them in the same direction. It is the most tedious part of the process, but it's important. You can use dedicated packages. Some of them help you with the harmonization, some don't. And we were able to check and see that actually, no, there was not an effect using this method. So using genetics to proxy how drugs might have their effect. This is now a standard part of drug development pipelines. Just because you can do something doesn't mean you should. This is where biology is very important. Recently, I was asked to review a study that looked at coffee consumption and whether your coffee consumption influenced type 2 diabetes. There is a genetic study that looked at which genetic variants are associated with coffee consumption. However, we tick the box, strong association, yes. Do the genetic variants only influence type 2 diabetes through coffee? My major question was, did you adjust for how much sugar people add into their coffee? Do you adjust for how many chocolate biscuits somebody has with each cup of coffee? For me, that's quite a few, so that'd be an important consideration. If you don't have that kind of information, if that information is not involved in the underlying GWAS on which you base the genetic part, you can't really tell whether or not the association is real. So you have to be able to consider, or you have to consider the original study. So do be aware of the phenotype and the covariates used in the analyses that you're basing your polygenic score on. Do match the ancestry of the genome-wide association study and the PRS. Do check the assumptions for MR analyses as much as possible. Do consider the biology. It's best if you can use a mixture of individual level and summary level data. Not always possible, but if you can, try. You can't use the same data for the GWAS and the polygenic score. It gets very circular logic. And don't forget how biased current data is. Most genetic data reflects 8% of the global population. That's not great. So always be aware that we are limited in what we can say at the minute. Thank you for listening. And here are a bunch of papers that are brilliant, are explaining the basics, the rules, and why, and some examples. And I probably talk far too fast because that's only 20 minutes. But so, any questions? If the people online have questions, either shout out or type in the chat.
Can I ask something?
Yes.
In the problem you mentioned before with the coffee and the diabetes, if we do a mediation analysis for the sugar, for the cookies, for something,
Yeah.
will it be enough?
If you do the mediation analysis for the underlying GWAS, yes, then you should be able to write that. You should be able to say you've accounted for that in the original study. When it comes to publishing, you probably have to justify that quite well.
Yeah.
But yes, as long as you're clear about what you have or haven't included and how that might have the effect on the results, that's fine.
Yeah.
So the study I was asked to review, they didn't mention sugar or biscuits or anything in the abstract. I'm like, well, it's such an important factor, you should have it in the abstract. If you haven't considered that at all of the assumptions fall apart. So yeah, whatever you find is then pretty meaningless.
Should they use the sugar GWAS? In this case, whether the sugar GWAS should be independent of the coffee and consumption GWAS. I mean, that in this case you have a like exposure and mediator sample overlap, and then usually, I mean, the researcher they would rule out the possibilities of the exposure and outcome sample overlap. But when it comes to like multivariables medianization, it is not always the case because it's pretty hard for you to find, you know, several GWAS that is independent of each other.
Yeah, for sure. So multivariable MR, I haven't gone into much because the last time I looked at the methods, there were some questions as to whether it was robust or not. So I kind of left it to one side. In that case, my suggestion would be do the MR for coffee consumption versus type 2 diabetes. Do the MR analyses for sugar consumption versus diabetes. And then see if they're the same, then it might be the sugar that's driving the coffee one. If they're very different, then maybe they have done a good job of separating out the coffee from the sugar.
In the same GWAS population.
If it's the same GWAS population, I mean, if you can get separate ones, that's great, but it's probably not possible because, say, UK Biobank is quite rare. That includes most people, yeah. So yeah, a lot of these methods are not perfect, which is why it's good to do a couple of different types of MR and look for consistency in the results. If you try publishing just one method of MR, you're going to have a lot of trouble with reviewers. So yeah, a bunch of different MR methods and look for consistency. It's the way forward. Desmond says, I believe if you have logistic regression GWAS, then the weight should be log odds ratio, not odds ratio, if you're making a PRS. Excellent point. Yeah, correct, Desmond. Thank you. Weighting by beta. So the log odds, not the odds ratio. Thank you. Any other questions? No more online. Okay, thank you very much. That was shorter than expected.
Got a question?
I think it's lunchtime. Oh, sorry. Did someone say something?
Yes, I've got a question. Desmond Campbell.
Yeah.
Oh.
Hi.
Basically, I think the strength of MR is that you've got, well, 20, maybe different 20 or plus genetic variants, and then therefore you can then kind of look for, well, you've got a distribution, so you get a distribution of effect sizes, and you can look for outliers, and you can throw them out and things like that. Whereas if you just do an MR with a PRS, then you've just got one, you've just got one effect size estimate, and then
It's true. I guess it depends on what your question is to some degree. Yeah, PRS, where you cluster all the variants before you put it into the analysis, whereas MR, you kind of put all the variants into the analysis together, and then you can do leave one out type of methods to see how stable the results are. Yeah, it's a good point. There are cases where using the PRS itself can be useful, especially if you're looking into, say, clinical translation of methods. Then building an algorithm that works with one PRS rather than lots of genetic variants might be a practical way forward. Might not.
sure thing.
Thank you. My email is on the slides. If you have questions, feel free to give me a shout. I might not answer immediately, but I will get back to you when I can. Okay, thank you very much.
Thank you.